Good afternoon. It's my great pleasure to introduce today's speaker for the G. Burroughs Miter Lecture, Steve Holland, known well to most of you, I think, in the audience. The Miter Lecture recognizes the contributions to the NIH of Bo Miter, G. Burroughs Miter, who was a director, scientific director at NCI, and then became director of laboratories and clinics from 1960 to 1968 at the NIH, which is the predecessor to my position. So I'm particularly fond of this lecture. And the other reason I'm particularly fond is that we get to honor one of our own, an intramural scientist, and not somebody that we brought in from the outside. And today's speaker is really a special honor to introduce, Steve Holland. He's the chief of the Laboratory of Clinical Infectious Diseases in NIAID, and he's my deputy for intramural clinical research. He was born in Buffalo, and he thinks the weather out here is summer. He got his undergraduate degree at St. John's College with an MD at Hopkins, did his residency and internship in internal medicine at Hopkins, where he was an ID fellow, and then later joined the faculty as an assistant professor. Then he came to the NIH, where he was a research associate in the Laboratory of Molecular Microbiology, also in NIAID. Then joined the tenure track and became a tenured investigator in the Laboratory of Host Defenses. For the past 10 years, he's been chief of the Laboratory of Infectious Diseases, and for the past 2, he's been deputy director for intramural clinical research. Steve's work is focused on phagocytosis and defects that lead to susceptibility to infection that affect the phagocytes. These studies have combined microbiology, genetics, physiology, molecular biology to define a set of rare diseases which have led to a much deeper understanding of how the normal body rejects infectious organisms. You'll hear one example today of a particularly interesting disease that does affect sensitivity to infectious disease. He's the only person I know of who has 4 NIH Directors Awards for his scientific research. If you go to the award ceremonies, you notice that most of the awards are for administrative achievements, not scientific ones. Reading these awards gives you some flavor of the breadth of Steve's work. He's co-discoverer of our very own bacterium, Granulobacter bethesdensis, which I call Steve's bacteria. He got an award also for his studies on the etiology of Job syndrome, for his description with others of the GATA2 deficiency, and also discovery of PLAD deficiency. He's an inspired and inspiring speaker, as you'll hear, and is frequently invited to give grand rounds and special lectures to audiences around the world. One of his jobs as Deputy Director for Intramural Clinical Research is to be an ambassador from the NIH to convince people that there's fun life at the NIH in clinical research. His work has been recognized with many awards, including the ASM Abbott Award, election as an NIH Distinguished Investigator, election to the American Academy of Physicians, among many other honors. So please join me in welcoming today's Miter lecturer, Steve Holland, one of our own, who will talk about the Protean, I love that word, Protean manifestations of GATA2 deficiency across the lifespan. Steve.
Thank you, Michael. Thank you, colleagues, friends, collaborators. There really is nothing more terrifying than having to speak here in front of people for whom I have such respect and with whom I work so closely. Not because I have any discomfort about the topic, but because your skill and your participation is so critical to everything that I do. And it is really a very humbling experience to present in front of you. Now, let me just start out by saying that I'm sure that many of you have heard too much from me this year. And I have been at several of these lectures, and if any of this is boring or repetitive, I can only say that I think the chairs here are quite comfortable, and I hope that you will enjoy them. But I'm going to try and tell you a comprehensive story about a disease that we've worked on for quite a while. And so, despite the fact that Les Biesecker told me today that I was inappropriately using the word protean because he has a patent on it for the Proteus syndrome, I'm going to keep on going, assuming that we can be okay. Now, I need to remind you that the complexity here is how do we recognize phenotype and how do we go from phenotype to genotype? And so it's always good to remember that phenotype has, like so many things, a Greek root, which is really about showing the type, showing something external. And as this particularly climate-appropriate cartoon shows, that really can be confusing. And of course, it's very confusing if you're one of these guys, and it might be rather attractive if you're this guy. But it's very important to keep in mind that what looks one way on the outside might not always be that way on the inside. So I wanted to take a little walk together down our path coming to the recognition of this disease over the last, literally, over 20 years. And this story begins, to the best of my knowledge, in 1989 with this paper here from the New England Journal by Christine Byron, who is on our Board of Scientific Counselors, reporting a very remarkable young woman who had severe herpes virus infections and then developed a fatal myelodysplastic condition, who had no natural killer cells. And this paper actually was brought to my attention. I was a fellow at the time by my father, who was interested in cancers, and thought that this disease with no natural killer cells must be very important in understanding cancer. And what was interesting about this young girl, the paper I had read at the time, but I must say I filed it away and didn't go back for quite a while, she had had an episode of life-threatening chickenpox, and chickenpox or varicella pneumonia, which is quite severe and can be fatal. And after she recovered from that, she had severe leukopenia, very low white count, that did not get better. And over the course of several years, she remained ill and subsequently died while trying to get to a bone marrow transplant. And the critical element in the paper reporting her in 1989 is shown here. These investigators were looking at the ability of natural killer cells to lyse virally infected targets. And here you see different, or to lyse cellular targets. Here you see controls that are able to lyse targets very effectively. And here you see cells from the patient, which were not able to lyse the targets at all. And so they came away with the take-home message, you know, NK cells are important, and they are involved in viral protection. The next year, a group that engaged Stu Orkin at Harvard recognized in chickens 3 proteins that were in hematopoietic and other cells that were all structurally similar. And these cells, they pointed out that these 3 proteins were different transactivating factors, but they all recognized virtually the same technique, the same sequence in cells, and they sometimes were within the same cell. And so they were puzzled by the fact that there were 3 proteins that were all recognizing the same motif. The following year now, so we're still in 1990, they further characterized this gene and this protein and recognized that it bound a very small sequence, and here in gels that no one in the last 10 years has ever run. They recognized that the gene sequence it was targeting was the same one that we now refer to as GATA, or sometimes they refer to it as TATC, just to keep us on our toes. And so they recognized a protein, a series of proteins that recognized the GATA, or the GATA sequence. And then finally, in 1992, they recognized that this same gene they now refer to as GATA2 was critical for activating the production of pre-pro endothelin. That is, this gene was critical for turning on the ability to make endothelial cells activate and proliferate. So here we have two very disparate things, severe viral susceptibility and then endothelial function, all being regulated by a protein. And obviously I'm telling you the story because these turn out to be all one thing. In 1992 then, the same year that they identified GATA2, I joined John Gallen's laboratory and began looking at patients with mycobacterial disease. And we identified a few patients. We started in 92. You can see I was much faster back then. We got this published in 1994. And what we recognized was that we could treat patients with disseminated mycobacterial disease with interferon, and it cured their infections. And here we had a little boy who had disseminated mycobacterial disease, and when we gave him interferon, he grew and cured his infection. And here a man who had recurrent abdominal accumulation of ascites fluid that was infected. When we gave him interferon, his ascites disappeared, and he was cured of his infection. And so we started accumulating patients who had these infections, and we started trying to treat them with interferon and to identify their underlying causes. The good news is we've now identified all the patients in that first report, and one of them, patient number 5, is shown here. So out of our sequence of patients with severe disseminated disease, the 5th one to come came in because she had severe disseminated Mycobacterium avium, and you see here a large skin nodule which was draining pus full of mycobacteria. And she also had a headache and complained of her left eye not working properly. And that, in fact, was a smooth muscle tumor that was infected with Epstein-Barr virus. Her story was worth noting because she had been well until she got into her 30s when she developed a pneumonia, which was treated with a lobectomy. I'm not saying that that's the indicated therapy for pneumonia, but that is what she wound up with at the time. Subsequently, she developed disseminated refractory mycobacterial infection despite very aggressive, very good therapy. And when she got referred here in 1992, we realized that she had no monocytes, no B cells, and no NK cells in her peripheral blood, despite having normal immunoglobulins and relatively normal T cell numbers. She also had severe perineal warts, but we treated her disease, her mycobacterial disease, with interferon, and she cleared it completely, went back to being a very healthy homemaker. But after several years, she developed progressive accumulation now of the monocytes. She'd had none. She now had some, and she developed chronic myelomonocytic leukemia. Well, we realized that this woman, patient 5, and another had very few monocytes in their peripheral blood and very few B cells, despite which they had adequate numbers of histiocytic, that is macrophage, and plasma cell infiltrates at inflammatory lesions. So our patient who developed the chronic myelomonocytic leukemia because back in the 1990s, bone marrow transplantation was still a field in its infancy, and we were not able to do her disease here, which I think was unfortunate for her and for us. She was transplanted elsewhere and died shortly after transplantation from a community-acquired respiratory virus infection. But she came back for an autopsy. We were very grateful to her and her family for being committed to our scientific enterprise. And it was clear that despite the fact that she had had very few monocytes in the peripheral blood, she had abundant macrophages in the tissue, and despite the fact that she had no B cells in the peripheral blood, she had abundant plasma cells in the peripheral tissues as well as normal levels of immunoglobulin. So the patient did not do very well, but in the process of evaluating her for transplant, we examined the rest of her siblings as possible donors, and we noticed, here's our patient number 5, that 3 of her sisters were healthy. One of them was not, and this woman had had severe warts from early in life and had no B cells, no monocytes, and no NK cells in her peripheral blood. In the process of taking the story from her, we also realized that her mother had had a virtually identical disease, beginning in her 30s to get ill, and in her 40s and 50s, she developed a syndrome that evolved into myelomonocytic leukemia with disseminated mycobacterial disease, and she died from that. The sister of our patient was seen here when she developed disseminated mycobacterial disease. We treated her, we cured her of her mycobacterial disease. And in the process of evaluating her, we looked at her chest CT, and you can see here, this is not a normal looking chest CT. Although it has, the lungs are well inflated, there's too much marking in the periphery. And there are too many small nodules here that had us concerned. And we offered her a lung biopsy, and she said, you know, I'm really fine. Why would I want to do that? I live in Denver. I don't have any problems breathing. I think I'll pass on your generous offer. And that was fine, and she did very well for several years, but then came back in 2007. And at this time, she didn't look as good. And you can see here now has severe lung disease. And although we thought at first that this was infiltrative, in fact, it turned out not to be. What this is pulmonary alveolar proteinosis, an abnormal accumulation of surfactant within airways reflecting macrophage, pulmonary alveolar macrophage dysfunction and inability to digest and degrade pulmonary accumulation. The standard treatment for this is whole lung lavage, which was performed in her and in several patients like her. And what you see is the standard sort of appearance, that is that the first liter is very opaque when much material is being recovered, and then over the time of now here 18 liters of lavage, a process that Ken Olivier, our pulmonologist, refers to as just as important as watching paint dry, but in fact is critical for clearing out pulmonary secretions and getting lungs back to normal. And what you see here is the material in the first one now being eluted out and cleared by the last one. So despite the fact that this woman too had no monocytes in her peripheral blood, she had lots of alveolar macrophages, and you can see here her pulmonary alveolar macrophages chock full of surfactant and other respiratory debris. So we recognized this syndrome beginning in 1992, but we did not recognize what caused it. And we knew that some of it was familial, we knew that some of it was not. We suspected it ought to be a single gene, but we were really confused about what was causing it. And so we went with a phenotypic description and referred to this as monomac for monocytopenia and mycobacterial disease. And it was characterized by abnormalities in these cells, both myeloid and lymphoid, that were absent from the peripheral blood. They had disseminated infections with mycobacteria, histoplasma, as well as many viruses. They had periods of aplastic anemia, but many of these patients then rapidly developed myelodysplasia with either acute or chronic leukemias. Several of them went on to develop pulmonary alveolar proteinosis, and when that developed, it was often associated with severe pulmonary hypertension. Now, some of the appearances here, just to sort of give you an idea of both how severe and how subtle some of this could be. Here you see extensive warts on the hand of a 24-year-old woman. Here you see an unsuspected hepatic tumor in a 25-year-old man, which on biopsy stains positive for Epstein-Barr virus, not in B cells where we expect it to be, but in fact in smooth muscle cells, indicating that he's got a problem in control of EBV in general and control of EBV in the proper tissues. Here is a specimen from a man who had a diffuse abscess in his psoas muscle, and on aspiration you see here this macrophage chock full of histoplasma and numerous organisms on stain. And then finally, some of these patients, especially before we had gotten our program fully integrated with that of Dennis Hickstein and the Cancer Institute so that we could move to bone marrow transplantation early, some of our patients died while we were trying to get things ready. And in this particular case, you see a man who developed the first recognized infection with the mold Neosartoria uragawa, which is in the family of Aspergillus fumigatus. And then, because phenotype is complicated, once you get into families, you need to look hard within those families. Here, this is in the man who had the disseminated histoplasmosis. His mother was otherwise asymptomatic in her 60s, except for what you see here, unilateral lymphedema and scattered warts along her leg. And these are the flat forms of human papillomavirus, which are more consistent with immunodeficiency. So after 18 years of studying this disease and not sure what the gene was, we decided we had better try to put out a phenotypic description in order to make sure that we cataloged what we knew and in order to begin recruiting more patients. And so the first 18 patients are collected here, and you see simply a phenotypic collection of what we understood. We collected patients based on their mycobacterial infection, but many of them had viral infections as well as other hematologic and other infectious complications. You also note that over the period we studied them, we had more than a quarter of patients dying. So a high mortality, a high rate of complication with a very confusing phenotype. Now, in the process of putting together their clinical phenotype, we were very focused on their laboratory aspects as well. And although I'm sure this is too small to read from the back, the point to take home is that all of these patients had abnormalities in monocytes, B cells, and NK cells. That is, over 2 different lineages, that is, lymphoid and myeloid, and variable abnormalities in their T cells. And when we looked at their bone marrows, what we saw was that they had a mixed picture of hypocellular bone marrows, as you see here, as well as very atypical cells, especially in the megakaryocytes. And this picture, courtesy of Kathy Calvo, shows the really unusual appearance of these megakaryocytes that are abnormal because their nuclei are too far spread apart. They're not developing normally, they're not working normally. And when Kathy does flow on these bone marrows, you see that not only do they have hypocellularity and atypical megakaryocytes, the other cell lines are abnormal as well. So this is the appearance of the granulocytes in a normal marrow. You see that they've got normal side scatter and granularity. And in patients who have advanced defects in this disease, monomac, their neutrophils do not have normal granules, suggesting that this is a defect that begins very early in myeloid ontogeny. And in addition, you see, just as we see in the peripheral blood, monocytes are absent in the bone marrow, and the number of lymphocytes is diminished. We can understand a little bit further some of what happens here, how it is that patients can have no monocytes in the marrow, but macrophages in the periphery. That's because macrophages can exist autonomously and replicate over years. And how can they have no B cells in the peripheral blood and yet make plasma cells and have immunoglobulins? And what you see here is that in a normal individual, mature B cells are easily detected, as are immature B cells, and you can see a progression of B cell maturation from CD10 positivity to CD20 positivity, winding up with a large, significant population of mature B cells, but also immature B cells that are developing. In our patients with this syndrome, though, this process is disordered. And what you see is that the early B cell precursors, hematogones, are absent, and that the B cell maturation is impaired because now immature B cells are gone. All that's left are a small number of the mature B cells. So these are the cells that presumably are involved in plasma cell development and in the ability to make immunoglobulin, and they are no longer being produced. So there are not going to be any early B cells available to respond to new antigens. So we've got a very complicated disease. It's got lots of manifestations, and here in a drawing that tries to capture everything, and of course therefore is very confusing, I've listed only here in red the very few complications that today's patient number 5 had: viruses, lung disease, disseminated mycobacterial infection, severe warts, and then developing myelomonocytic leukemia. But you can see there are many other manifestations, and these are manifestations of exactly the same disease. So how can we put together this complex a background, that is, with cellular defects, infection susceptibility, bone marrow dysfunction, lymphatic dysfunction, as well as lung dysfunction, all of which is in an autosomal dominant pattern. Therefore, it really has to be a monogenic disorder. And I could tell you about all the ways we missed this for all those years. 20 years working on this disease. It is that expression, NIH stands for not in a hurry. But it was an opportunity to collect the information and the phenotype. And then after going down many of these blind alleys, Amy Sue in the laboratory said, hey, wait a minute. How can you possibly involve all these different cell lines? Megakaryocytes, neutrophils, monocytes, dendritic cells, B cells, NK cells. The only way you can achieve something that gets all of those cells is to have something that's affecting the hematopoietic stem cell. You must be at a much more primitive precursor, and that precursor ought to be at the hematopoietic stem cell level. And that started us down the track of looking for transcription factors in the hematopoietic stem cell, and Amy carefully set out what are all the possible targets and then went about sequencing them. And to cut through further problems, I can just tell you that it was on May 3rd of 2011 that Amy called me early in the morning exclaiming 8 out of 8, 8 out of 8, that we had identified 8 out of our target patients with mutations in this gene, now known and known earlier to others as GATA2. And why should GATA2 be the right target for this? Because it fits all the criteria. And let me just point out that what it does, like many transcription factors, it controls other transcription factors, GATA1, GATA3, and others. It controls histone deacetylases, which are involved in chromatin winding and unwinding, endothelial nitric oxide synthase, which is involved in endothelial function, as well as angiopoietic factors, which are critically involved in the development of endothelium and lymphatics. So this gene really does have it all. We identified that on May 3rd. We were very lucky to find sympathetic editors at Blood who were able to get this paper out 5 weeks later, because we thought it was so important an observation, and they agreed. And it was a wonderful example of how working with colleagues at the publication level, which is a critical component for all of us, when they understand what's important to us and we understand what's important to them, everyone seems to do very well. And at the same time, simultaneously and subsequently, this gene was identified in several other syndromes. And so the group of Hamish Scott and Marshall Horwitz had been looking at familial myelodysplastic syndrome and leukemia. And I had become aware of their work, which helped us to know that GATA2 was a good candidate. And they also identified this gene in some 4-generation families, and they thought that this was a syndrome of out-of-the-blue leukemia. That is, all people got was myelodysplasia leukemia with no other problems. The group of Matthew Cowan in England was very interested in dendritic cell deficiency, and so they plowed through a series of samples and recognized people who had no peripheral blood dendritic cells who turned out to have exactly the same problem that they called DCML for dendritic cell monocyte B and NK lymphoid deficiency. And then the group of Sahar Mansour in England noticed a syndrome that is referred to eponymously as Emberger syndrome, which is the syndrome of lymphedema and myelodysplasia or acute leukemia. And you see here an example of the lymphedema associated with myelodysplasia. And subsequently, we were able to work with the group of Hamish Scott and Marshall Horowitz and others to identify that the reason that the lymphatic dysfunction is there is not only the abnormalities in pre-pro-endothelial expression, but it's the expression of GATA2 in the lymphatic ducts themselves. And as you see here, here is a lymphatic valve. Here is the expression of GATA2, which is critical for lymphatic development. And then, of course, finally, we went back when we recognized that GATA2 was involved in NK function and viral susceptibility and could be both sporadic and familial. I contacted Christine Byron, who said, I don't have any cells. I contacted John Sullivan at UMass, who had a vial of peripheral blood mononuclear cells stored from this girl from the 1990s, and we were able to go back and identify that she too had a mutation in GATA2 that led to a frame shift and a stop. Now, you may recognize the problem, one set of foundational documents, many different interpretations of them. And how do we go from one to many and from many to one? So there are many ways to call this disease. We started out with monomac and our colleagues with these, but the key point here is that all of it is due to GATA2 deficiency. And I think as we move forward, the name that we should use for this is simply GATA2 deficiency and not our other more eponymous names. So how does this work? Let me just remind you for a minute, because when we identified GATA2 as the responsible gene, I had absolutely no clue why this would be important or how it worked. But this is one of those two-digit zinc finger transcription factors that Winston Churchill first described. And you see here the way that they impact. You have both fingers that are straddling DNA, and then they have domains at the C and the N terminus that recruit other transcription factors into the complex to turn on and turn off the respective genes. And they bind to the sequence here of GATA or TATC, giving them a target. There are 6 mammalian GATA factors, and they sort of break generally into the hematopoietic and the somatic group. The hematopoietic group include GATA1, which is involved in an anemia, especially associated with Down syndrome. GATA2, which we're talking about. GATA3, which the lymphomaniacs think is important in Th2 control, but is actually clinically associated with a disease called HDR, hypoparathyroidism, deafness, and renal disease, and is not associated with any recognized immunodeficiency. The GATA factors 4, 5, and 6 involve many other targets that lead to atrial and ventricular defects, coronary and urinary tract abnormalities, as well as pancreatic disease. Since this is the depiction of the zinc fingers in GATA2, in the GATA factors, and what you see in red are the completely conserved elements. So what you notice is that all of these are essentially the same in their zinc fingers and are therefore able to bind to exactly the same targets, giving you some sense of why the phenotype might be so broad. That is, GATA2 is going to be able to bind to GATA1 sites and vice versa, and it stands to reason, though has not been yet proven, that GATA2 can bind to GATA4 sites and vice versa, which might explain why there is an overlap in some of the structural aspects of this disease, along with the immunologic ones. Now, one of the things that makes this gene so complicated is that it's not only involved in the regulation that I've talked to you about, but it is not involved so much in binding to promoters, which is where I think many of us started out our sort of molecular careers thinking that activity was going to take place. Most of what the GATA factors bind to, and GATA2 in particular, is intronic and in enhancer regions. That is, outside of the structural elements that are directly associated with gene regulation as we think about it most commonly. So now we've got a problem. We've got heterozygous changes in GATA2, and they are associated with disease. We know that they are monogenic because we can see them travel in families. And so now we've got to figure out how do they function. We get 3 choices here. They could be dominant negative, dominant gain of function, or haploinsufficient. And this has been a slightly controversial point, but I'm going to use the clinical epidemiology to try and make, I think, the persuasive point that there's only one mechanism that can possibly explain it. And the simple thing to observe is that when we have gone through now and looked at all of the patients with this disease, we find three classes of easily identified mutation. Missense mutations, which are typically in the second zinc finger, in which a protein is produced. Nonsense mutations, which are spread more broadly, in which no protein is produced. And intronic mutations, noted here, which are quite interesting because almost a quarter of our patients have these intronic mutations. And this intron 5 is where GATA2 binds inside its own gene to upregulate itself. Now, the important point is that almost all of the factors, all of the patients who have any of these mutations are virtually indistinguishable. That is, their clinical phenotype is almost identical across all three of those. Making it even more persuasive, there is another small group of patients in whom we can't find a mutation, but what we do see is that only one mRNA is produced. So one allele is being expressed, one allele is not. And as far as we can tell, these patients are all the same. And we can see this clinically because the disease that occurs, here I've just taken myelodysplasia, no matter what kind of mutation they have, the rate of progression and the development of disease is the same. That is, no matter whether you're making protein, not making protein, expressing an allele, or only expressing a little bit of allele, they're all the same problem. And so this must then reflect that the underlying mechanism is one of haploinsufficiency. That is, one allele is simply not enough. And we can prove this at a molecular level by looking at the number of GATA2 transcripts that are present in cell lines. And here you see that no matter what the underlying mechanism of the mutation, whether it's intronic or heterozygous, missense or nonsense, they all have the same consequence, which is low expression of GATA2 compared to the normals and compared to other patient populations. So haploinsufficiency is the organizing theme behind these problems. Now, what are some of the aspects of this disease that I think would be useful to think about as we move into the later aspects of understanding it? So we've got dominant transmission because of allelic loss leading to haploinsufficiency. What are some of the consequences? There are many things that I think we're going to learn out of this one disease. One of them, so far, has been something about NK cell biology. So I started out telling you about a girl with NK cell deficiency who turned out to be GATA2 mutated. GATA2 is required for the expression of the CD56 bright cells, which are so important in NK function and biology. And these cells are absent in GATA2 deficient patients, leading to impaired ability to perform cytotoxicity, antibiotic-dependent cell-mediated cytotoxicity, as well as several other critical functions for NK cells, presumably leading to their susceptibility to viruses and to cancer. We found this underlying defect in several other hypoplastic marrow disorders. So in about 10% of juvenile myelodysplasia, those are due to underlying GATA2 mutations. In 5 to 10% of juvenile aplastic anemia, those are due to underlying GATA2 mutations. In the French series looking at pediatric neutropenia, 10% of those are due to GATA2 haploinsufficiency. So there are going to be many different manifestations. One of them here is illustrated by this case, a 12-year-old boy with severe aplastic anemia who came in for evaluation and treatment. He received standard therapy for his aplastic anemia, which was successful, except that 2 years later, after his treatment, he came back with severe myelodysplasia and monosomy 7 and rapidly evolved into an acute myelogenous leukemia. Only after his death did we recognize that his underlying mutation was an intron 5 mutation in GATA2. Some of the other manifestations that are important and what make this disease so difficult to have recognized at the beginning are viral infections. So here we're plotting the percent without a particular manifestation. So early in life, up until about age 10, children are normal. But following that, they develop viral infections, typically with human papillomavirus. And you can see that by adulthood, all of them have some HPV manifestation. Mycobacterial and fungal infections occur a little bit later and progress over time. So in our series, granted we recruit for these infections, but in our series, patients develop more mycobacterial disease and fungal disease over time. And then myelodysplasia, which comes on around the same time and following the development of the severe infections, also runs a progressive course over the course of GATA2 deficiency. Following these things, I think reflecting progressive, severe pulmonary alveolar macrophage dysfunction, pulmonary alveolar proteinosis becomes a major issue, as I showed you in our first patient and as seen in this one. So, cataloging disease is important for science, and it's very important for us as investigators. But patients need something more. And you cannot simply say to patients, boy, this is great. We now understand why you're going to die. That's just not an adequate answer, and that's not why we have a hospital. If that's what we wanted, we would just be a research institute. And so it's critical to have collaborators who are as motivated and as aggressive and as interested in the disease and its treatment as all of us are in disease and its etiology. So it's been my great good fortune to collaborate with Jennifer Cuellar-Rodriguez and Dennis Hickstein, who recognized in this disease a fantastic opportunity to identify something novel and to treat it and to come up with critical observations that would be different from all observations in previous episodes of bone marrow transplantation, because when you go into a new disease, you've got new things to do. And so I just take one figure from their paper, which illustrates that after transplantation, exactly what you'd expect happens. NK cells come back, as do monocytes. That is, this means that the problem is not in the stroma of the marrow, the problem was in the hematopoietic cells themselves. That can be replaced and that can be repaired. In addition to that, not only are the hematopoietic elements repaired, but you can repair end organ damage as well. So in this, our first patient who underwent bone marrow transplantation here, his lung disease was so severe, he was on 6 liters of oxygen and on high-dose full-time Viagra to try and control his pulmonary hypertension. It was a dicey thing to take him to transplant, but lucky for us, we had an aggressive bunch of transplanters who realized that there were only 2 choices, progress or death, and thank God they chose progress. And with transplant, what you see is that over 6 months, he was able to come off oxygen and come off sildenafil and recover normal lung function for a time. He's had subsequent problems and has had to undergo lung transplantation. But from the hematopoietic standpoint, he was able to make it through. Now, the last thing I need to tell you about is that what we recognize is disease, and that is what comes to us as clinicians. But once we recognize a gene, then we have to go back and recognize people before disease develops. And how we understand that then leads us to the next steps. So here I've just plotted, this is some work that Michael Spinner and Lauren Sanchez did, pulling together all of our data on all of our patients and looking at B, NK, monocyte, and CD4 numbers. And here in the numbers below are simply how many disease manifestations people had. And it doesn't matter which one it is, but if you had no disease manifestations, the key point is you got normal numbers of B cells, normal numbers of NK cells, normal numbers of monocytes, and normal numbers of T cells. As you accumulate disease manifestations, everything goes to hell. And so the question is, why? Are people getting sick because they are cytopenic, or are people getting cytopenic because they're sick? And this is a critically important question because it drives what we decide to do in terms of prophylaxis, in terms of therapy, in terms of preemptive transplantation. So one piece of information about this was just published last week from Emery Bresnick's group at University of Wisconsin, a tremendous bunch of collaborators for us, in which they were able to make heterozygous mice, which they've been good enough to share with us. And what you see here is when they take embryos from these heterozygous mice and look at the number of stem cells that they can transplant, when you do serial bone marrow transplants, you see that in normal mice, the transplants are able to grow successfully and expand. When you take heterozygous mice, they have trouble expanding. And in competitive transplantation experiments, they don't compete very well. And if you have complete knockout, those don't make stem cells that are able to do anything. And these animals are able to undergo, the wild-type animals are able to undergo secondary transplantation to transmit again, but the heterozygous knockout animals do not do very well, indicating that these animals have diminished numbers of stem cells at the beginning, and that as time goes on, their stem cells remain impaired. You can see this when you try to culture stem cells from these animals. And here, the wild type has, this is the normal level of CFU-GM. And here in the heterozygous knockout animals, the number of CFU-GM is significantly, about 50% reduced. Well, how does that help us? So now we need to go back and do some of our bench-to-bedside experiments, recruiting willing volunteers, as we so often do. Getting consents administered has been a bit difficult, but thank God we've got a very good IRB. So our approach here is to take our heterozygous knockout animals and infect them intravenously with M. avium complex and to ask what happens to their bone marrow. Can we reproduce the problems in the knockout animals that we have in our humans? And so what's important to know is that at the beginning, when they're not infected, the bone marrows are completely normal in the knockout and the wild-type animals. And so here, when we are looking at our GATA2 uninfected, these are wild-type animals on the top and mutant animals on the bottom. When they're uninfected, here on the this part of the screen, when they're uninfected, the number of stem cells in the bone marrow is, detectable and just like it ought to be. These are reduced compared to these, but they're still present. But once you infect the animals, you see that the wild type do not lose significant numbers of stem cells, whereas after infection, and not before, the GATA2 mutant animals become stem cell depleted. And we can see this here when we look in the flow cytometric study. If we look at a very stringent marker for murine stem cells, the heterozygous animals after infection are profoundly depleted, whereas the wild type are not. Therefore, suggesting that it is the infection itself that is helping to drive the cytopenia and not the cytopenia that is making the infection occur. This, I think, is important for informing us in what we should do about prophylaxis and in terms of what are the kinds of things that are likely leading to their problems. Okay, so lastly now, we've talked about germline problems in GATA2 with mutations that are present from birth and that move on. But GATA2 is not only involved in that. And so in pediatric acute leukemias, the development of GATA2 mutation occurs in the setting of pre-existing CBP alpha mutation. And in that setting, GATA2 mutations seem to be ameliorative and to have somewhat better survival than in patients who have CBP mutations alone. But if their GATA2 is being overexpressed, so in these patients it is normally expressed, if GATA2 is overexpressed in leukemic cells, that leads to worse prognosis. So there's having a normal thing, a good thing, and then there is, of course, too much of a good thing. So in conclusion, how does GATA2 do all this? It has regulatory effects in numerous tissues. Abnormal vessels and lymphatics overlapping targets of the GATA factors, which I think is a critical point in how we understand how diffuse its mechanisms are. Early population of tissues with macrophages and plasma cells, with then depletion of monocytes and B cells, leaving those cells stranded out in colonial sites where they get no replenishment and no nourishment. Impaired definitive hematopoiesis, infection-driven bone marrow exhaustion, and then secondary mutations and cytogenetic effects. So GATA2 deficiency turns out, I think, to be much more common than we thought early on. It is involved in many different diseases with multiple manifestations that cut across disciplines. It predisposes to a host of infections, which makes it difficult to anticipate. It's got a variable onset, but then progressive cytopenias. Its mechanism is haploinsufficiency with regulatory mutations indicating that the control of this gene is extraordinarily tight. And finally, it's already taught us a lot about NK cell deficiency and dysfunction, and I think it's going to teach us a lot more about cancer susceptibility in the future. So this is one gene that ties together infection, hematopoiesis, immunodeficiency, lymphedema, pulmonary alveolar proteinosis, and cancer. And there aren't many of those. So I've tried to take you on a trip, tried to start you with the first recognition of this disease, inchoate though it was, in 1989, and walk you through the 22 years to recognition of the genetic basis of this disease, and then tell you a little bit about the still evolving and complex manifestations of this condition that are still being worked out now. So I like to think of this as the syndrome of the blind men with the elephant, and GATA2 really is whatever it is that you want it to be, depending on where you're standing. I started out here thinking that it was mycobacterial disease, but I've learned a lot about its HPV manifestations, its lymphedema, its fungal manifestations, and the myelodysplastic and acute leukemic problems, as well as the lung disease. Of course, what makes it difficult in terms of genetic counseling and understanding in the long run is the fact that at some point in everybody's life, they're normal. And how to anticipate that, how to work with it, what to do about it, is really what the next phase of our studies is about. So phenotype, it changes with time, it changes with exposure, it changes with environment. Whether you get satisfaction or not, you really still have to be thinking about what is it that I expect at this time and what is it I expect this disease to do in this person. So with that, I'd like to conclude by acknowledging the extraordinary number of people that have been involved in this project from its get-go. And in particular, Amy Sue and Elizabeth Sampaio have been critically important in the laboratory aspects. Lauren Sanchez and Michael Spinner have pulled together all of our clinical data. Kathy Calvo and Diane Arthur and Mark Raffeld have been critical molecular and hematologic collaborators. Stefania Pitaluga has helped us with all of our immunohistochemistry, and Li Ding has been driving our mouse work and keeping that afloat. Many other collaborators here internally, some of whom I mentioned as we've gone along, and our collaborators in the NCI, without whom we would have only a program that would be able to say, I'm so interested in your problem and I'm so sorry for your loss. And if we do not maintain an institution that insists on moving beyond the cataloging of disaster, we will not have a future that is exciting in which to live. Thank you very much.
I'm sure that Steve would be happy to take questions. And while you're getting to the microphone, let me start. Since this is such a Protean syndrome, have you looked at, or has anyone looked at polymorphisms that occur in the normal population? And do any of them represent a spectrum of disease that might be interesting?
Yeah, that's a very important point. The spectrum of disease associated with polymorphisms is not this set of diseases. In fact, there is a SNP association with cardiovascular disease, which I don't understand, and that's the only one that I'm familiar with so far. Right now, though, a lot of people are looking in leukemias and other myelodysplastic syndromes, and I think we'll come up with a broader spectrum both the mutations and probably of some SNPs. And I think as the whole exome approach is more exhaustively explored, I'm sure we'll find other associations as well. Dr. Hickstein.
Steve, I'm wondering if how well that mouse model recapitulates the human disease. You know, there's a lot of concern about mouse models in humans, and the mouse has been around for 25 years. Those hets have been running around labs, and nobody ever saw the humans and thought, oh, gee, there's a mouse just like that.
Right. So, how good is the mouse model? We had hoped it would be a little more better, and we've been a little disappointed that it took 22 weeks to achieve a stem cell depletion in the mouse model with just mycobacterial infection. I think one of the problems is knowing whether we're challenging the mouse with the right thing. I chose MAC because that's what gets the humans. Well, mice don't get MAC. In fact, mice are very hard to infect with MAC. So what we have to do is to go back now and think about what's the right thing to put into the mice and what should we do about that as we go along. Whether the mouse will be a good marker for human disease, I think, is a tough question. Lisa McReynolds in the laboratory is working on crossing it to other known gene defects so that we could see if we could accelerate some things like, in particular, myelodysplasia and leukemia. And those would be critical targets to know whether GATA2 is interacting with those. We'd like it to be a better model.
You mentioned that GATA2 and GATA1 overlapped in terms of their binding capabilities as well as GATA2 and GATA4, yet you had sort of distinct clinical syndromes associated with them. Do you think there might be more overlap between those syndromes, or I guess how do you explain why the syndromes, even though GATA2 has such Protean manifestations, how they're still just sort of different types of syndromes?
Yes, I think that's a very important point, Jeff, and I think that there are a lot of overlaps in the syndromes, and the overlaps occur at the level of, I think that the number, we are limited by what we have studied, not only us, but in the other GATA factors. And although they are recognized as having Mendelian associations with many of these things, they are not numerous, the case reports. And just as Marshall Horowitz had published his patients as having out-of-the-blue leukemia, in fact, now that we've gone back and met his patients, it wasn't out of the blue and it wasn't only leukemia. But we're all subject to the questions that we ask. We ask infection questions, they asked cancer questions. We all got our answers, but it turns out they're all in one whole. The overlap of symptoms with GATA1 is very important in terms of the thrombocytopenia and the anemia that these patients get. With GATA3, sensory neural hearing loss is a critical part of GATA3 deficiency, and it's a very common manifestation of GATA2 deficiency. Now, proving that the defects, that the overlapping syndromes are because of overlapping binding sites is a little bit difficult. What we can show with some confidence is that the targets overlap and that the binding can be coincident. And what's sort of exciting about the initial recognition of the chicken with those 3 GATA binding factors all in the same cell is that they knew they bound the same targets, but they didn't know what the level of complex regulation was. Just last week, another paper in Cell Stem Cell has looked at the fine-tuning of GATA binding sites, and although GATA is the canonical site, there is some subtle variation when you look at GATA1 and GATA2 and GATA3 in terms of what they do, and it varies from tissue to tissue and time after stimulation. So I think that there will be a complex set of manifestations that we look forward to working out. Dr. Waldman.
As this disease evolves into myelodysplastic and then leukemia, does it share with run-of-the-mill myelodysplastic and polycythemia vera a JAK2 mutation at 267 that's being treated with ruxolitinib now?
Not that we've seen so far. We've not looked for JAK2 mutations in all of our patients, but in those in whom we have looked, we've not seen it. I think the mechanisms are likely to be different, but I think one of the critical things we need to do, and we've not yet done, is to undertake very aggressive sequencing at multiple stages along the development. So Rob West in Dennis Hickstein's lab has looked at the accumulation of ASXL1 mutations and shown that those are a very important precursor to the development of leukemia, especially CMML in these patients. The JAK2 mutations I have not seen yet. I don't know, Rob, if you've looked in any of those patients. It's an important question, and we don't have any proof of that yet, but I think as we do more whole exome and whole genome sequencing down the road, we ought to be able to see what the spectrum of mutational development is. We'd like to be able to understand what it is that triggers there's trisomy 8, monosomy 7 are very common events here. We'd like to understand the genetic and genomic instability that seem to be part of this.
Hi, very fascinating talk. What are your thoughts about why it takes so long for the manifestations of GATA2 mutation to become manifest?
Yeah, so this is a very perplexing problem, and I guess I have two and a half answers. One is, life's like that, and sometimes we see diseases in which there's a lot of phenotypic variation, and that may have to do with a variety of environmental factors. Number two, which I think is what we are trying to focus on, is how much of this is infection-driven. That is, if we can identify infections that are making some people get sick earlier and prevention of infections that help some people to get sick later, we would like to identify that and like to try and prevent those. And then, of course, number three is that there might well be levels of genetic variation within families. And so some of the family clusters in which we've had wildly spread onsets of disease, even in people growing up in the same house, et cetera, suggest that we need to go back and look at regulatory factors that might be turning on or turning off GATA2. Along that line, the fact that regulation is so tight and that having only 70% expression of the gene still gets you sick, that means that the critical 30%, that gets you between sick and normal is an area in which there can be a lot of intervention. And so thinking about how we can upregulate GATA2 expression, maybe therapeutically, is really something that we'd like to be able to focus on. So maybe there are genes that are giving some people slightly higher levels and others slightly lower.
Hello, Dr. Holland. So always when you have GATA2 deficiency, is it directly correlated? It is obvious that NK deficiency and dysfunction, always it happens in every patient, or is
well,
there
the NK dysfunction has been seen in every patient at every time. NK deficiency, that is low number, has not always been seen. So this suggests that there is a separation between NK function and NK number. And in some of these patients, especially those that have persistent NK number, we can upregulate their function with interferon alpha. And we've used this therapeutically in some of the patients who have bad viral infections quite successfully. In terms of what drives why it is that NK cells disappear over time, that I don't know. Why they start out normal and then become low with time, I can't tell you that yet.
And I have another quick question. So is there any particular ethnic background or demographically in some parts of the world they have GATA2 deficiency, or it's not discovered yet? Yeah.
So I think, as is true for many severe diseases that are often fatal, they don't last long in populations. And so, yes, we find some families in which we've got 3 or 4 generations, but we've seen so far, no ethnic predisposition and no regional predisposition. Now, this disease is still in its very early days, and the fact that we see the same mutations arising at the somatic level in leukemias and at the germline level in our patients tells you that these are spots that like to get mutated. So it's certainly possible that there are some ethnic groups that might be more prone to mutate those regions because of some other mutator factors, but so far we haven't seen that.
So I'll ask about autoimmune disorders associated. The CD56 bright NK cells has been called by many immunoregulatory NK cells, making IL-10 and control. And Bibi Bielikova, in diclizumab treatment of multiple sclerosis, felt that the amelioration that it provided was an increase in this subset of NK cells. In their absence, is there any kind of autoimmunity?
Yeah, that's a very important problem. And the autoimmune features of this disease are detectable but not dramatic. And it's also hard to know, in the setting of complex multi-tissue gene expression defects, what's autoimmune and what is intrinsic dysfunction. That being said, hypothyroidism is relatively common in this disease without previously recognized antibodies. Doesn't mean there aren't some, or some infiltrate. We've not seen much in the way of local inflammatory involvement. So hypothyroidism, paniculitis is very common in this disease in people who have had disseminated mycobacterial infection, even when we can't find the organism. Whether that represents a local inflammatory dysregulation or an incipient infection, which we cannot identify, I don't know. There have been other examples that were called primary biliary cirrhosis in this disease, and I understand as much about it in this context as I do in the general context. Finally, some of the inflammatory reaction has been quite dramatic and severe, which might get at these issues about counter-regulatory influences. In particular, Clostridium difficile infection has been dramatically severe in this disease. We've had one patient die, and one had a colectomy, and another one wound up with, on a ventilator because of C. difficile infection, even in children, which is virtually unheard of. So there's something that these people get infected and then have a more dramatic process, even with something that is limited to the luminal region. And that might be something that's at the level of a dysregulated function. We don't have the kinds of tissue samples that would let us go back and look at NK infiltration and say whether that is up or down. Thank you very much.
Steve? Thanks for a terrific talk. Bo Miter would be proud. Let me remind you that there's a reception sponsored by the FAS in the library. You're all welcome. And you can ask Steve some more questions then.